I should get started. My name is Jian Yang from School of Life Sciences here at Westlake University. First, I'd like to thank Catherine and Andrew for providing me this opportunity to present here today. So my group has been focusing on genetics and genomics of complex traits in humans, and traits that are common, and most of the traits in our population we see are complex. Including human height, obesity, heart disease, cancer, and mental illness. So I guess many of you know genome-wide association study as a technology. So just for completion, very briefly, GWAS is a technology where you can collect data from a large population and use throughout high throughput genotyping technology to genotype all the individuals and then associate each individual genetic marker or variant to a trait of interest and run a test, one genetic marker at a time, for evidence of genetic association with a trait of interest. GWAS has been criticized for not being able to identify many genetic variants. So here's an example to show that in 2009, a case control study with a few thousand cases and controls for schizophrenia was not very successful. It didn't have power to identify any genetic variant parts passing the genome-wide significance level. And now the concern is more about that there are too many variants being identified. And here in this example, again, for schizophrenia, when the sample size increased to about 77,000 and over 243,000 cases controls, then the overall number of genome-wide significant loci for schizophrenia have passed over 280. So now the problem is, just give you another example that we actually Dr. Longda Jiang, then he was in my group, demonstrated in similarly for body mass index using the UK Biobank data. So in this experiment, Longda took 1,000 unrelated individuals from the UK Biobank and didn't really identify any locus passing the genome-wide significance level. So then if you increase the sample size to 10,000, you will see one hit here. This is actually the first locus identified by GWAS in 2007, the FTO locus. And if you further increase the sample size to 100,000, you'll see about 50 loci, and further to include all the unrelated individuals available in the UK Biobank, you'll get about 500 loci, which in total only explain about 4.4% of the BMI variation. So this is a clear demonstration of what is now called as a polygenic model. So the reason why the number of genome-wide significant loci increases with the sample size is because there are actually many genetic variants, each explaining a small fraction of a phenotypic variation or heritability, and because of their the effect size for most of them are small to be detected if the sample size is not large enough. And governed by this equation here, while the expected value of the chi-square test statistic is approximately 1 + 2 p is the allele frequency, 1 times 1 minus p times n is the sample size, while b is the effect size. So if the effect size is small, you will need a large sample size for common variants. But if for rare variants, P is small, so still you will need a very large N, even if B is relatively large. So why is that? Why do we have such a polygenic model for most traits in our human population? So here is a kind of one hypothesis to explain this polygenic pattern of inheritance. So from the work from us and others, we show that there are two possible reasons to explain the polygenic model. One, there must be a large number of mutational target genes, because otherwise you won't see such a large number of variants. And why the effect size are all typically small for common variants. So we believe that this is very likely to be due to purified selection, where in the evolutionary process, new mutations with larger effect will tend to be more likely to be eliminated by purified selection, or also called negative selection. So variants with relatively small effect size can become common variants because of genetic drift, and those with relatively large effect size are typically kept at a low frequency as rare variants, and in the population we see in the contemporary population. So here's actually a review paper from Patrick Sullivan and colleagues to demonstrate clearly there's no signal in this area where you have large effect size and also high minor allele frequency or high. So the only exception is ApoE, the ApoE E4 allele in Alzheimer's disease. And certainly there's a power issue, but clearly this shows that you only possibly see large effect for rare variant. And this is actually a consequence of negative selection. So I think the next, identifying genetic variants associated with a particular common traits or disease is not a big problem, and we have efforts keep increasing the sample size to gain power to identify more variants. The problem now is how can we go from genetic association to genes and then to mechanisms, and further to translate these findings into clinical applications and medicine. So why it is so difficult to identify the causal genes underlying GWAS signals? So here are a few plausible reasons. The first reason is that the mapping resolution for current GWAS is not great because of LD. So in this case, you will have a GWAS locus where there are many genes underlying, so the actual causal gene is not clear in this case. You may also see that the resolution could be higher in some locus like this, but they are often located in non-coding region. So often not clear what are the causal genes. The leader variants may not be the causal because of imputation, error, sampling variance, etc. But even if we know the causal variants, it is not straightforward to link between causal variants or variants and genes. So this now being an area of sort of very intensive research. It's a concept to integrate a GWAS and EQTR data. Well, EQTR is a concept that gene expression level, difference in gene expression level among us can be associated with genetic variation. You know, in the typical GWAS design, we are actually testing a difference in phenotype among genotypes, genotype groups. If we know that the gene expression level is also associated with genotype, then we can possibly combine the two data set to see if gene expression is associated with the phenotype. So there are actually many methods for this kind of integration, and we have contributed one piece called SMR. Though the concept behind SMR is very simple. So we kind of adopt the idea of Mendelian randomization, and the effect size BXY represents the association or the effect of gene expression on the trait, which can be computed from the ratio between the GWAS effect size and the EQTL effect size. And the standard error for this BXY head can be also approximated by the GWAS and the EQTL standard error and effect sizes. And these analyses can be done using summary statistics only, and those GWAS and the EQTL data can be from different samples. So here is just a demonstration to show why can we pick up a plausible or putative causal gene from a GWAS signal. So here is analysis team just run today for this presentation. So team took the PGC data, the latest data published in 2022, and uses the data she generated with it's kind of a meta-analysis of multiple brain EQTL data sets. So so in this locus, the furin gene is picked up as the likely causal gene, and the reason is that the EQTL signal for furin is in high agreement with the GWAS signal at this locus. And we have expanded the SMR to be able to analyze other kind of molecular phenotypes. Here in this case, demethylation. And here is a very interesting example that I often would like to talk, is that this is a type 2 diabetes GWAS signal. This SNP is a lead SNP in this area. So if you look at the nearest gene, you may think these genes are actually closer. But if we integrate the EQTL data, we'll see that CAMK1D is likely to be the putative causal gene here, and supported by two probes. If we then look at the EQTL result for CAMK1D, you see it shows a nice colocalization with the GWAS signal. Very interestingly, if we annotate this area by the epigenome roadmap project data, you can see that there are a few DNA methylation sites also showed association with type 2 diabetes around the promoter area of CAMK1D. And if you look at these methylation QTL signals, they also show there are very high agreement with the EQTL and the GWAS signals. So given this kind of information, we can hypothesize that possibly the SNP located in a kind of cell type specific or tissue specific enhancer area with this allele, T allele and the C allele substitution, we may see a difference in binding affinity of an activator. So because of the, we can tell the direction from the SMR effect size. So we can hypothesize that this is likely to be activator. And because of the difference in allele, the binding affinity of the activator to the enhancer will decrease, and therefore the methylation level on the promoter may increase. That will further decrease the expression level of this gene. And that mechanism has actually been approved by a study 1 year later by wet lab experiment. So SMR has also been applied in many different cases. And here is just an example that we applied the Yang Wu applied the SMR to the COVID-19 GWAS data generated by a project led by Kenny Bailey. So there are actually a number of GWAS hits for COVID-19 susceptibility or severeness. TYK2 is actually the highest hit after integrating the EQDL data with the COVID-19 GWAS data. So TYK2 is actually one of the 4 gene targets for an existing drug called baricitinib. So Kenny and colleagues put this drug into clinical trial for COVID, and show actually a sort of a reduction in mortality with this drug in COVID. So we've talked about the use of EQTL data to prioritize genes from GWAS signals and the possibility to integrate other omics levels. So by definition, EQTL is a kind of association between genotype variation and the amount of overall RNA abundance. So while in this case, you can see that there are 2 genotype groups. In this case, there's no difference in overall RNA abundance, but there's a clear difference in isoform composition, suggesting that there could be a variant affecting RNA splicing, therefore will lead to a difference in isoform composition. So we developed a method called CSO to scan the genome for this kind of genotype and splicing association. So we applied a CSO together with another splicing detection method called leaf cutter to this data set with over 2,800 brain samples from over 2,400 individuals. So very interestingly, we see a lot of genes with splicing QTL. We define this variant associated with splicing as SQTL. We have many genes with SEQTL only, and those genes are called S genes. And certainly we have genes with EQTL only, and there's a substantial overlap. If you look at the overlapping genes, you see that it's about a half the EQTL signals and the SQTL signals for the same gene are actually in very low LD, suggesting there are separate signals. And this is kind of confirmed by, if you run a colocalization analysis, also about a half of the EQTL and SQTL signals are shared. So if we then look at the EQTL, and the SQTL, then clearly the SQTLs are enriched in genomic element related to splicing. And we further asked how many or how much proportion of the GWAS signals can be explained by SQTLs in addition to EQTL. So we run EQTL and the SQTL in the same data set and then integrate the SQTL and EQTL summary statistics with the GWAS summary statistics for about 12 brain-related phenotypes, all with very large sample size. So interestingly, we kind of using SMR and colloq, this kind of method to integrate GWAS and EQTL and SQTL data. You see that the number of genes prioritized using SQTLs only is even larger than that using EQTL only, with a moderate or overlap. So here just give you a simple example here. This is again a GWAS signal for schizophrenia. If you look at the EQTL signal here, there's no EQTL signal supporting this GWAS signal. But if you look at the SQTL signal from CSO for DGKZ gene, and there's a nice support here. So we have quite an amount of requests from SMR users in terms of formatting the EQTL data, the different kinds of XQTL data, and a lot of data analysis questions. So then we decided whether there's opportunity to develop an online analysis tool where just to make the data analysis a lot easier. So we developed this SMR portal where users only need to upload a GWAS summary statistics. Although we also provided an option for users to upload their own XQTL data, where XQ, I mean any molecular phenotype uses as a trait for GWAS. And certainly those EQTL or XQTL data you uploaded will not be shared across users. So once you create a job, you will see from this task list a list of tasks you have already submitted. And if the job is completed, you will get an email notification. And once you receive an email, you click the link, it will direct you to the task list page again. And here is a visualization button. If you click here, it will direct you to a page where all the prioritized locus will be shown. And if you click any of the locus, you will make it will generate a plot like this for you, with on the GWAS signal on the top, followed by XQTL signals from different omics level, tissue, or data sets. And we will also provide you the epigenome annotations with the gene underneath. And given the data we've curated, we have run this kind of SMR analysis with over 200 traits together with over 100 SQTL data sets, and put that all these kind of associations into a database for you to search. And if you type a disease or a gene name, you will get a list like that, and you click any of them. We also provided tools for you to do this kind of visualization. All right, but the problem is for the SMR or SMR portal, even with many XQTL from different omics levels, we still have to run the analysis for one data set at a time. So here is a method we developed a couple of years ago called Opera, the idea is to integrate, to do a kind of joint analysis of one GWAS data set with many XQTL data sets from multiple omics levels simultaneously. Certainly, it doesn't have to be a multi-omics level. It can also be applied to multiple tissues. So here is an application. The purpose of this analysis is trying to, at least at the time, collecting the most powerful XQTL data, like the PQTL data, EQTL data from EQTL gene, and methylation QTL data, splicing, polyadenylation QTL, histone modification, and chromatin accessibility EQTL. And integrating these 7 XQTL data set with the most powerful GWAS for 50 traits, I think we find some encouraging sort of results. Is that with all 7 data sets, we found about 50% of the GWAS loci can be mapped to at least one molecular trait. And of course, there are cases where a GWAS signal can be mapped simultaneously to multiple molecular traits. But still, with this 50%, the rest of 50% is still missing. So we kind of hypothesize that there could be context-dependent genetic effects not being accounted for in this analysis, because we use the XQTL data typically from bulk data, and we don't have separation of cell types. So to run a cell type dependent genetic EQTL analysis, we will possibly need to do cell sorting or single cell sequencing, or a more cost-effective approach would be cellular deconvolution, where you can deconvolute or estimate cell type proportion from bulk sample and then do an interaction model to see if the EQTL or XQTL effects are cell type dependent. There's also a possibility that genetic effect may vary not only among cell types, but also different cell states within a cell type. So with single cell data, we can now be able to estimate what is so-called pseudotime or cell state trajectory for a particular cell type. One particular, a typical example is a T cell. We have naive, activated, and exhausted states. And these states can vary under different biological conditions. But those kind of single cell data sets are still of relatively small sample size. We know that we have a lot of bulk analytic data. So can we estimate or deconvolute some sort of distribution of cell abundance of cells at different states, or the cell abundance versus cell trajectory distribution? So the strategy is that we can possibly get estimate the cell state trajectory from a small single cell analytic data set, and of course with tissue, gender, and population matched with the bulk data. If we can map this bulk data to the single cell data, we may possibly be able to estimate the cell state abundance distribution for each individual in the bulk analytic data. So we developed a method called Medusa. So the idea is actually also very straightforward. It took marker genes for a particular cell states trajectory. Once we have the marker gene, we can link the the gene expression level in the bulk tissue and the gene expression level it can essentially be a single cell, but for the ease of, or for robustness, we take cells of similar states as a kind of being, we call the focal states, and this regression coefficient is actually an estimate of cell state abundance. To improve this the accuracy of the model, we also model other cell types as a fixed effect in this case, and also all the remaining cells as random effect to explain more variation from the model. So we run some simulation to show that the model performs well regardless what kind of distribution will be. And actually, in fact, when the distribution is complicated, this medusa shows actually even more advantage over existing method. And we have also benchmarked this method by real data set where you have single cell RNA-seq data and the bulk RNA-seq data applied to the same samples. And in that case, we can estimate the cell state trajectory from single cell RNA-seq data and do deconvolution, and see the consistency between the two estimates. And in this case, medusa also shows higher performance than the other methods. So with this a high accuracy method for cell state deconvolution, we made an effort to do this kind of cell state deconvolution, in this case, epithelial keratinization trajectory. So we apply that to the GTEx esophagus tissue, around 500 people. And once we have a deconvolution of this cell state trajectory distribution for each individual, and we can see that for each individual there are some variations for this curve. So if we divide this distribution into 4 quartiles, you see that and use that the abundance in each quartile as a phenotype, essentially as a variable. And for interaction analysis, the phenotype will still be the gene expression level, while the cell state abundance in each quartile will be the variable for interaction analysis. So on the right-hand side, you can see that we detected a number of, we call it cell state dependent E genes. For those genes, we detect a significant interaction effect between genotype and the cell state abundance. So then we further to validate whether we've really identified something that is cell state dependent. So we use the esophagus single cell ataxic data from another data set and estimated this epithelial keratinization trajectory, and then look at those chromatin, open chromatin marks that is associated with this trajectory. We call these are the sort of cell state dependent open chromatin region. Essentially, the marker open chromatin marks for this trajectory. And then we tested whether the cell state dependent EQTLs are enriched in those cell state dependent open chromatin regions. We actually found that this is the case. So with the bulk EQTL, there's no enrichment here, and there's a clear enrichment of the cell state dependent EQTLs, and that enrichment increases with a higher level of statistical significance. And then we further integrated the cell state dependent EQTL data into GWAS and identify a couple of GWAS signal that is co-localized with the cell state dependent EQTLs only. Here's some examples of 2 dietary traits from the UK Biobank. And interestingly, in this case, actually roughly the same, I would say the same locus for different traits, pleiotropy effect. And these 2 signals actually not co-localized with the bulk EQTL signal, but with cell state dependent EQTL signal. But of course, we also have cases, and this is the GWAS for gastroesophageal reflux disease, where we identify a couple of GWAS signal, although not passing the genome-wide significance level in GWAS, co-localized with both bulk EQTL and cell state dependent EQTL. All right, so now we got many different XQTLs, right? So we have even for 1 omics level for the transcriptomics, we will have EQTL, SQTL, and we can have cell different tissues, and we can have different cell types, right? And we will also have different omics levels like the proteomics and the further epigenomics features like demethylation, chromatin accessibility, and histone modifications. So how can we possibly integrate everything all into one test? So we've discussed, we mentioned OPERA, but still OPERA has a cap. So OPERA would work with like less than 10 XQTL data sets. Beyond that, OPERA will become very slow. So we were thinking whether we can develop a method that has no limitation in terms of number of data sets that can be included. So Ting developed this method called MAGIC. It's a very quick and test. So the concept is that we kind of put everything into a gene-based test. So for gene features or transcriptomic or proteomic features, you can run a kind of gene-based test. But for the epigenomic features, we first use linkers to link like a chromatin interaction and similar technologies to link epigenomic features to a gene and then further integrate everything into a gene-based test. So we have applied this method MAGIC to a very large number of XQTL data, over 400 XQTL data sets from 7 omics levels, including both bulk and cell type, even single cell, or I would say bulk and cell type level EQTLs, even including data from different developmental stages. So with MAGIC, we can run this kind of analysis with all these data sets together. And with the GWAS data, we picked 11 quantitative traits and 46 disease traits, all with large sample size. And now the bottom line is that we can map. Remember, this number was 50% for OPERA. And now with a very large, a lot more XQTL data, different omics layers, and both bulk and cell type level, we now identified 87% of the GWAS signal are mapped to at least 1 XQTL. So the gap is actually closing. I think I'm running a bit slow. This is actually the last piece I want to talk about, is that we can possibly use GWAS to identify the relevant biological context for a complex trait. So we will have a method previously to identify tissues or cell types that are relevant for a trait. And with a single cell RNA-seq data, we can even identify cells or groups of cells that are relevant to a trait. But how about the spatial spatial transcriptomics data? Or can we possibly do a spatially resolved mapping of cells to complex traits? So we've developed a method, well, actually a project led by 2 students called GSMAP. So it's a method to combine GWAS data with spatial transcriptomics data. So the idea is actually very simple and straightforward. First, we identify a marker genes for a spot in a spatial transcriptomic data. But because of, for some spatial transcriptomics technology, the sort of the number of genes can be captured by a single spot is limited. So to improve the robustness, we used a graph neural network to integrate cell gene expression and the spatial coordinates into a single embedding vector, and then use cosine similarity to measure the similarities or the among spots. The aim is to identify spots that are similar in cell type and their spatial location. So once we identify a friend, I would say friend spot, you can use a friend to help the focal spot to get a good estimate of the or good ranking of the marker genes. So with that, once we have the marker gene for a spot with the help from neighboring spots, we then ask whether SNPs around these genes are enriched for a heritability for a particular trait. So in a way to link a spot to a trait. And the strength of this heritability enrichment can be measured as the strength of a relevance between a spot and a trait. So here is an application to demonstrate how the method works. So we took the data, so we don't have at this stage, we don't have many data from humans. So we started with mouse. So here is a mouse is a mouse embryonic spatial transcriptomic data published in 2022 using the StereoSeq platform. So then we mapped 3 traits: IQ, hemoglobin concentration, and height. So you can see that clearly. IQ was mapped to the brain and the spinal cord. Hemoglobin concentration was mapped to almost exclusively to liver, while height was mapped to largely mapped to cartilage and its primordium. It kind of confirm our prior knowledge, right? And we further run simulation to show that if we don't have simulated traits from real genotype data, but without some sort of allow given different settings, we don't see any inflated false positive rate for the mapping. So kind of so the previous examples can be used as positive controls, and here are the negative controls. So so we not just use this method to demonstrate the positive controls. Here are some new novel discoveries. So in this case, we map male pattern baldness to the mouse embryonic data. So we have prior knowledge, certainly epithelial cells are most likely to be the relevant cells. And so in this case, we labeled sort of 6 areas where the epithelial cells are distributed. If we do the mapping, only L2, the L2 area showed the very strong association. So what are these cells? If you look closely, the L2 region, this is actually the face region for the mouse. These cells are not randomly distributed. They distributed like the hair follicle cell assemblies. We further confirmed by the hair follicle marker gene that these cells are indeed the hair follicle cells. Okay, so the mouse in embryo is still too early, right? And then we mapped some human brain related traits to the mouse brain spatial transcriptomic data. So here is kind of a half brain from adult mouse and at a single cell resolution actually. So when we do the mapping for the brain related traits, we included traits like intelligence, schizophrenia, depression, bipolar disorder, all brain related. So interestingly, cortex shows the highest relevance to most traits. And the second highest associated region actually vary across traits. For example, hippocampus CA1 is the second highest association for IQ and schizophrenia, and midbrain show a very strong sort of association with depression. All right, so here is another kind of positive control. We also included a Parkinson disease, but if you look at the lambda GC for the GWAS summary statistics, you can tell that the GWAS is less powered compared to the other traits. But still, we were able to identify just one significant hit here. So this is a brain region called substantial nigra ventral tegmental area, a region known to be relevant to Parkinson's disease. This region is kind of is enriched for dopaminergic neurons. Again, I would say another positive control. So how about a comparison? Can we compare the sort of among different traits? So to remove or consider the difference in GWAS power, we define a odds ratio. Essentially, it says strength of association with the brain region relative to the whole brain. So it's kind of relatively is a strength association of a trait with the brain region relative to the whole brain. Okay, so for we compared schizophrenia, depression, and IQ. So schizophrenia showed a very high sort of relevance to hippocampus CA1 and CA2. But depression shows kind of depleted association in the hippocampus region, but a high enrichment in the midbrain. So midbrain is a lot more relevant to depression relative to compared to schizophrenia. And IQ is something in the middle. What's even more interesting is that if we just look at the glutamatergic neurons, which shows the most abundance in the mouse brain, at least in this hippocampus CA1 area. If we just look at the glutamatergic neurons, we see that the kind of the strength of association is not evenly distributed. It is actually spatially patterned. If we plot the strength of the association, each dot is one single neuron, one single glutamatergic neuron, and on the x-axis is the distance to the dorsal side. The dorsal side is this side. The closer the distance to the dorsal side, the strength the association is. And then we asked, why is that? So we then, because we have the spatial kind of coordinate, right? So we can actually identify genes that are associated with this spatial distribution, or we can also do a association of gene expression level with the strength association. Both results are highly consistent, pointing to a few genes that are kind of related to calcium signaling and regulation. So if we then do the enrichment, so the geo-enrichment of those genes, and they are also enriched, kind of enriched in calcium transporting signaling related pathways. All right, so mouse is probably too different from humans, right? So we further applied this GSMAP to macaque cortex data. So because of the limitation of current spatial transcriptomic technology, we don't have the data from the whole brain. We only have data from the cortex, again, from a publication from a different group. With this macaque data, we have different, we have kind of technical replications and also replications among different animals, and show the robustness of GSMAP. So if you look at this macaque prefrontal cortex, then actually they can be this region can be sort of classified into different subregions. And if we map those brain-related, some of the brain-related traits to that data, we see that depression show a kind of outstanding association with the 14R region. So what is the 14R? 14R is called the gyrus rectus. And this distribution, this association actually, or the glutamatergic neurons in this region actually also not uniformly distributed. And you look at this lateral to medial axis, the strength of association with depression actually increases. And if you look at the lateral side and the medial side showed a stronger association than the lateral side for depression. Okay, so let's recall the result for mouse, right? So we showed previously with the mouse data that midbrain is relevant to depression. And now we show that the 14R medial side showed also relevance in the macaque data. If you get the highly expressed gene from the macaque 14R medial region and the midbrain or CA1 region from the mouse, you see there's a strong kind of enrichment. And if you look at then the highly expressed genes in those two regions, one in macaque, one in mouse, those genes are actually enriched in biological processes related to neuroplasticity. And more exciting, at least to me, is that if we look at the target sort of genes that are highly expressed this in 14R medial area, and then do an enrichment of the drug target genes, the psychiatric drug target genes in humans, there's a very, very strong enrichment. So again, genes are highly expressed in 14R medial in macaque shows very high enrichment in psychiatric drug targets in humans. So it's suggesting that once you identify, if you can identify the most relevant cells or brain region for a psychiatric disorder, a lot of the highly expressed genes in those areas or in those cells will tend to show very high overlap with the causal gene for the disease. And finally, we show that although we don't have many human data set, there's one dorsolateral prefrontal cortex data from humans with slightly low resolution. And we compared this data with macaque data And although there's different, there's alignment issue and different technology, there's some level of consistency between the two data sets. So with that, I think I'm running over time a little bit. I'd like to finish off here by thanking all the people that have done great work in producing all the results I just talked about in my presentation, and a lot of the work that I actually have done by the past members of some of whom have established their own research groups. And I'm also grateful for funding support from the Westlake Educational Foundation and National Natural Science Foundation of China, and the Science and Science and Technology Department of Zhejiang Province. And thank you for listening, and happy to take questions. Yeah, so the question is about magic. We are pushing very hard to make just for your information. So most likely, because of the large number of XQTL data sets involved, most likely we will develop again an online analysis platform. So in that case, you will only need to upload your own summary statistics. And certainly we'll provide an offline version, but in that case, you will have to download all the XQTL data sets. There are many of them. Yeah, I don't have an estimate of the timing, but yeah, I'm pushing. We are pushing very hard. Yeah.